Click for IB Biology. We're looking at nucleotides. These are the monomers of the nucleic acids DNA and RNA. There's a blank PDF of this presentation down in the description. It's linked in there that you can use to annotate your own notes. And then there's a set of 10 questions, of course, with answers that you can use to test your understanding of the content of this video. At the end, there's some channel links to the next video in the sequence on polynucleotides, and also to the playlist and to the channel itself. Okay, so let's take a look at what we're going to be dealing with. So, nucleic acids, the two major forms that we'll consider are DNA and RNA. DNA is the main molecule of genetic information. RNA is better known for its role in protein synthesis and the regulation of gene expression, actually. When we deal with DNA, usually we're dealing with a molecule which is known as the double helix, which means that it's actually composed of two polynucleotide chains. So we're going to take that, we're going to polymerize that into a chain, which is the polynucleotide. But DNA actually has two of them, whereas RNA forms such as messenger RNA, transfer RNA, and ribosomal RNA, these usually are, these are always composed of a single chain. So these chains, whether they're double helix DNA or RNA single chains, are made up of the nucleotides. So let's take a look at those nucleotides, the monomers. So the monomers seen here in this chemical diagram have this PO4 group here. That's the phosphate group. And then here we have a pentose sugar known as ribose, and then here we have what is called the base. For the purposes of this examination and for these videos, students are advised to make diagrams using this representative shapes rather than to recall the details of the chemistry. That said, knowing that this is a 5-carbon pentose sugar, we can see that on the first carbon, 1', we have the base attached. 2', the second carbon, this is an important one because here in RNA, that carbon, the 2' carbon, will have an OH group and an H group, but in DNA, this carbon, the 2' carbon, will simply have 2 hydrogens attached, and hence the deoxy part of the name of deoxyribonucleic acid. So the oxygen is removed, the one which is present in RNA. 3', this is an important carbon to know for the process of polymerization. 4' carbon is here. Now we must recall that this is a pentose having 5 carbons, and so between the ring structure and the pentose is the 5th carbon, 5'. So we have 3' and 5' in these locations here, and that will become important in our communication about the structure of the nucleic acids. When we compare the nucleotides in DNA, then we find that there are 4 types of base. There is adenine and guanine. These have double ring structures and are known as purines. And then there is thymine and cytosine. These have single ring structures, as shown here, and are known as pyrimidines. So DNA will have the nucleotides adenine, guanine, thymine, and cytosine in its structure. When we come to RNA, this will also have adenine, guanine, and cytosine, but the thymine is replaced with uracil. So if we just make a brief note on that, so we have A, T, G, and C types of base, whereas RNA would have A, U, G, and C types of base. In the next video, we will look at how these monomers are joined together to form polynucleotides. There's a link here to the channel and also to the playlist.